All right, so watch and listen carefully here, guys. We'll go through the kind of pre-lab stuff here. So what we're going to be looking at tomorrow, okay, are now specifically the structures within a cell. Okay, so we're going to look at an example of a plant cell. Okay, that'll be the monocot dicot cross section. That'll be our plant cell. Okay, essentially what that is they took a leaf and kind of laid it on its side and then cut it so that what you see is the layers of the leaf. Okay, so not looking down on it from the top, but kind of holding it and looking at it on its thinnest dimension. Okay, so what we're going to be looking for in there, okay, we're going to be looking at the cells that are on the top layer of this because they're the most typical plant cells. Okay, they'll be nice and square. You'll be able to see a nucleus and chloroplasts and all of that kind of stuff. Right? We're going to be looking at a generalized animal cell. Okay, the name of that slide is going to be stratified squamous epithelium. Okay, which is the fancy way of saying that red skin you get when you dust your knuckles. Okay, doesn't bleed so much but stings like crazy. Okay, that's what stratified squamous. It's just underneath your outer epithelium layer. Okay, that's what the cells will be looking at. They're kind of all random. Okay, they're kind of everywhere, but there's lots of them, but they're quite small. Okay, so you do have to zoom in quite a bit, and I'll show you what all these look like here in just a few minutes. Okay, we're also going to look at a human blood smear. Okay, and the only reason we're looking at that is because everyone always asks. It actually isn't one that we're going to use to answer any of the analysis in this lab. Okay, the reason we're not going to look at it in terms of that is that red blood cells do not have nuclei. Okay, a red blood cell does not have a nucleus. Red blood cells, once they're produced, will not divide to reproduce. They divide from essentially bone marrow and stem cells and things like that in your body. But once they're produced, they don't have a nucleus, so they're not going to divide themselves. They're short-lived. Okay, their job is carry oxygen until you die. Okay, so we're just going to look at them because people are always curious what they look like. But we're not. We'll do a lab diagram, but they will not be included in the analysis portion. Okay, and the other thing we're going to look at is one of your own slides. One of your own cells slides. You're going to make a slide of one of your own cells. You're going to take a Q-tip, okay, and just like CSI, you're going to swab the inside of your cheek and you're going to smear that onto the same kind of slide you did the letter E on. Put a drop of water on that, a cover slip, just like you did with the letter E, okay, and on there will be a whole bunch of your own cells, okay, and these are usually the ones, even though they're not stained a color, they're usually the ones you can see the most structures in. Okay, if we fiddle around with the light, okay, on the underneath side of the slide, or sorry, the stage, there's a little lever that opens and closes this iris that's on the bottom. And as you play around with that, you can increase the contrast of what you're looking at, okay, which makes it look sharper. And it just suddenly they'll just jump out, and you'll see all these structures within the cells once you find a cell. Okay, the difficulty is finding the cells amongst all the debris that's going to be on that slide. Because no matter how much you brush your teeth, rinse your mouth out with Listerine or whatever, okay, you're still going to have pieces of food in your mouth. Okay, so if you're looking at something that's like big and thick and green, it's a piece of food. Okay, the cells are transparent. Okay, you have to get them pretty well to see them. They're not going to be really, really overtly obvious. So if you're looking at something that's really big and dark, you're not looking at one of your cells. You're looking at something you chewed but didn't swallow. Okay? Yeah. It's gross, but it's there. Okay? Don't be ashamed. No one's going to make fun of you except me. Okay. So those will be the three that we'll be using to answer our problem. Okay? The problem is, where have we seen this before? It's the cell theory. It's the second point of the cell theory, okay? We're trying to figure it out. We're trying to figure out what structures are necessary for cells to do this, right? So what we need to be looking for when we're looking under the microscope is what kinds of cellular structures do all of these cells have in common? Okay, so we got to look closely. We got to find things that are in both plant and animal cells, and then we also have to look for structures that may not be in all of them and figure out why they're important. Looking for things that would help a cell carry out its basic functions. And what are some of those that all types of cells would have? Which ones are specific? Okay, so your design, okay, manipulated, responding, and controlled variables. Okay. Obviously, we're looking at different slides under the microscope, but obviously you're going to need to be a little more specific than that. Okay, talking about maybe why we're looking at different cells and those different cells maybe being from different organisms. Okay, talk about that a little bit and kind of why. Obviously, our responding variable, we're looking at cells for a reason. There's something we're looking for. Okay, that's what you need to talk about for that. And then anything that's the same for all the times you look through the microscope. So a lot of these will be similar to your microscopy lab, but there's going to be one controlled variable that will be different. We're looking at cells from a plant, we're looking at cells from an animal, and a cell from another animal, you. Okay? Animals and plants all have a certain kind of cell that we named and talked about yesterday. Okay? That is something that will need to go in as a controlled variable. You will have to look it up, okay? But it needs to be in there as a controlled variable. All the cells we look at are a certain kind. Okay? All right. In our hypothesis, okay, I would say the if part of our hypothesis needs to talk about this. Okay. The and part of our hypothesis is just describing what we're doing. We're looking at a whole bunch of blank cells. Okay. And then the then part, you should probably tell me what things a cell is going to need to have. What are some of the structures you think all the cells you look at will have? And maybe what are some that are specific, maybe only to plants and animals? Okay? Tell me which ones you think will be there. Which ones are you realistically going to be able to see? All right? You're not going to see ribosomes, so you might as well not even bother listing it. They're going to be too small. You're not going to see them. Okay? All right, so same as last time. Once we get looking at the slides and you get a specimen focused really well, okay, you're going to take a picture of that, send it to your group mates. The pictures I've seen so far on the microscopy lab have been excellent. Okay, they've turned out really well. We had a few issues so far with format, however. Okay, if you recall, it was important on a lab drawing for all the labels to be horizontal lines going to the same side. I do have a lot of sunburst style lab drawings where labels kind of go everywhere. Okay. But that's really about my only big beef so far on those, has just been that. The rest of it's been good. They've been clear, okay, things like that. Now, when you label these diagrams, however, I'm looking for the real names now. We've gone over what the parts of the cell are. You've got to do a decent job of figuring out, okay, that furry thing looks like it could be this. Okay, that big green blob is probably this, that kind of stuff. Okay. You only have one analysis question. But that means I'm going to want a fairly detailed answer for it. Like I'm talking a paragraph that's decently long. Okay, maybe 2 paragraphs. Based on your knowledge of cell structure and your observations in the lab. Okay, different cells, what organelles are most important for a cell to function, and which ones are specific for a certain mode of nutrition? Explain. Okay, I'm looking for a fair amount of detail. I'm looking for a lot of good logical reasoning there. Okay, if you just write, I saw all these, they were everywhere. Okay, I'm not going to be very impressed. Okay. Got to have a nucleus. It's the brain of the cell. You'll get a big fat 0. Okay. Yeah. Come up with some other stuff. All right. Conclusion. Remember, restate your hypothesis. Just copy and paste it. Explain why, given what you saw in the lab, if it's right or wrong. Okay. And then obviously sources of error. Okay. And suggest improvements. Also, something, guys, in your microscopy lab, there was a lot of human error listed in the microscopy lab thus far, just on the ones I've looked at. Okay, don't tell me that the slides are dirty and we should clean them. Clean them. Okay, clean them before you look at them. Don't say, "Ah, well, it was dirty, but I didn't care. I just looked at it under the microscope anyway." Okay, no, you got to clean it off. Then that's not a source of error. You didn't do it. You knew exactly what to do and had the ability to do it, but didn't. Okay, that's like again saying, "Hey, I screwed up, but I just didn't fix it." Okay. Yeah, so come up with something other than sources of human error. I would have even accepted inexperience as a source of error. Okay? How many times have you honestly used a microscope in your life? Okay? Does that contribute to your ability to see things effectively? Certainly it does. Okay. By the way, you can't use that now because you've already had a lab where you got the practice. So okay, ruin that one for you. Okay. Well, no, because now you're practiced up. Yeah. All right. Questions on what's expected there? Okay. The things we're going to be looking at tomorrow. Okay. This is what you're going to be seeing. The first one here, this is the plant cell. Right? We are going to be wanting to do the cells up here, okay, on the top layer, right? Because they're the most stereotypical of plant cells, right? You can very clearly see things, okay? You can see lots of structures in here. They're kind of faded, but they're there, okay? There's lots of stuff that you can see in there, right? This is the stratified squamous epithelium. This is the animal cell. Now, when you're looking at this one, I'm going to blow it up a little bit more, okay? It's going to be really, really small when you look at it. This is one animal cell. Okay, the dark part in the middle is a nucleus, obviously. Okay, but you need to get one of those, like that, and they're not going to be very big. So I mean, you've got a lot of them to look at, but what I don't want to see is nucleus, cell membrane, like no, because then it looks like you didn't have any idea what you were looking at. Okay, you've got one. You want to be looking at one kind of individual cell. And once you get them blown up, I mean, you can see a fair amount of stuff. You can see there's some stuff in the corner of this cell here. Okay, you can see that there's some structures that seem like they're attached to the nucleus. I'm not saying use your imagination, but use your imagination a little bit. There's no other really way to say that. You have to look at it and kind of be objective and go, okay, there's usually this around there, so that's probably what that is. Be logical about it. Carly, question? Well, as many structures as you can. If you need to use more than one cell to do all the labeling, that's fine. Okay? But I would suggest that once you take that picture, you blow it up, a single cell as best you can. Blow it up and make it really big so you can get most of your labels from one cell. Okay? The blood's going to look something like this. Okay? The little purple ones here, those are just red blood cells, and then these are white blood cells. Okay? Leukocytes and lymphocytes and things like that. And then it'll be either orange or purple. It depends on which way it was stained. There's a few different stains that were used. These are my cheek cells from a few years ago. Okay, that's a cheek cell right there. That's a cheek cell. That's a cheek cell. These are cheek cells all stuck together. Okay, you can see a lot of structures in here when you get them blown up. Okay, they really stand out. When you get it blown up a little more, you'll be able to see. Yeah, but you can see, there's obvious structures in and around here that, you can at least use your reasoning to get a decent label on, right? So that's the stuff that you'll be looking at tomorrow through the microscope, but it gives you some idea what to expect. Okay? Now, you can see that this one's in black and white. It's not really in black and white, it's actually in color, but it looks black and white because there's no stain added to it. Okay? It's just going to be your cells, and there'll be a little fiddling around to get that one. So do the cheek last. Okay? Something to be cautious of, because I've had people do this in the past, and I want to make sure that it does not happen. Okay? Other than the cells from the inside of your mouth, you are not to look at any other types of cells from your body in the lab. Okay? Right? Do not prick your finger and get blood anywhere. Right? There's, obviously risk of infectious diseases and things like that that we do not want to be taking any chances with, right? That could be blood-borne, but would not be borne in saliva, okay? Things like hepatitis, HIV, things like that, okay? We're not allowed to take any chances. It's not that I think any of you would have any of those horrible diseases, but, okay, I'm not allowed to have any of those kinds of bodily fluids, okay, in the lab. Yeah, that's why I'm letting you look at the preserved blood slide. It's not your blood, it's safe blood, okay, and we don't have to worry about it. All right, so just please be mindful of that. Other stuff for tomorrow in the lab. I don't know how many of you noticed, but I always find it's difficult to look through the microscope if I'm wearing my glasses. I find it's easier if I'm wearing my contacts. Okay, so I don't know if you found that yesterday, if it bothers you or not, but if it does, know that we're going to be in the lab tomorrow looking through there. Other thing, if you're wearing, what's the stuff that makes your eyelashes thicker? Mascara. Okay. Sorry, I'm a guy. I don't know that stuff. That oftentimes will come off while you're looking in the microscope and you're blinking. It comes off on the microscope on that ocular lens, and it really makes them dirty and oily. Okay, so if you could maybe not put that on until after the lab or something. Okay, avoiding, yeah, no makeup day maybe. Okay, it does create a bit of a cleaning problem for us, so if you could avoid that, it would be much appreciated. Okay. All right, you have about 7 or 8 minutes to do some pre-lab stuff there on the Chromebook, so get her done.